Due to COVID-19, everyone should be familiar with the term PCR test by now. With its help, an infection can be detected. But how does it actually work? Stay tuned and find out why the PCR has not only been a breakthrough for diagnostic, but also for genetic engineering in the last decades. The PCR is a brilliant method to copy specific parts of DNA sequences. The method is orientated towards the natural process of DNA replication, which plays a major role in cell division. It ensures that every daughter cell contains exactly one copy of the cell's genetic information. Taking this process as an example, an artificial concept was born to amplify DNA sequences called PCR or polymerase chain reaction. But which steps does a PCR include, and how can it be explained on a molecular level? DNA consists of two complementary strands built from base pairs. These base pairs being complementary means that every base is only binding to exactly one partner in the opposing strand. Adenine is only binding thymine, cytosine is only interacting with guanine. This being said, the first step of replication, but also of the artificial PCR, includes the separation of this DNA double strand. Both of the forming single strands code for the same genetic information, which is why they can be used as templates for new DNA molecules. The separation is performed by heating the DNA sample, which breaks the bonds between the double strand's base pairs. For the second step of the process, it is necessary to target a specific sequence in the DNA. This is achieved by so-called primers. Primers are single-stranded, short DNA fragments that bind to the part in the DNA which shall be copied. This marks a starting point for a certain enzyme, the DNA polymerase. This enzyme is able to fill up the complementary bases and therefore creates two new double strands on the template of two single strands. An exponential duplication is achieved, meaning that with every cycle, the DNA strands are doubled. But how is this method connected to the detection of COVID-19? COVID tests are based on the detection of viral genetic information. If viral genes are present in the sample, we use specific primers for the amplification. In the COVID PCR, every amplification of the viral genes gives a signal. The higher the viral load, the stronger the signal. However, PCR is not only of particular importance for diagnostic, but also for every step performed in genetic engineering. PCR offers the possibility to amplify and modify even small amounts of DNA sequences and therefore contribute to genetic modification of several organisms.